Welcome technicians. Today we look into how to manage our microbiological section of our laboratory. So first and the foremost thing, identify the culture plates properly. So first one is the blood agar plate, McConkey agar plate, the Kled agar plate, chocolate agar plate, Muller-Hilton agar plate, and the UTI plate. Okay. So what are all the samples we normally receive? Sputum, bronchial wash, tracheal secretions. So many type of samples we normally receive. Okay, so for stool samples we use only McConkey agar plate, whereas for urine we use Kled agar plate. Okay, so for all others we need to inoculate in the McConkey agar plate as well as in the blood agar plate. Okay, so always this thing is first blood agar plate, then McConkey agar plate, and then you make the slides. That is how the inoculation process should go. Okay, so first and the foremost thing, take the blood agar plate, divide it into two, and put the barcode on the section which you are going to put to use. So always remember, if the pus culture or whatever sample is there, if you look at it is turbid or organisms are plenty or anything, don't divide the plates. Instead use the whole complete plate. Otherwise when you know that it is not that many, then you can use half of the plate. So it's a you have to know whether to use a single plate or a two in a single plate two samples. So that you have to decide yourself, or you can always ask the HOD. So you divide the plate into half. So put the barcode on the side which you are using, and then identify the this thing properly. Like for example, 11 to 4 o'clock, 12 to 6 o'clock, and 9 o'clock to 3 o'clock, like a clock. Okay, so put the inoculation from 11 o'clock to 4 o'clock position like this. Okay, don't try to make round like this. Instead you go in this fashion. Okay, properly you go in this fashion. Make sure that there is a plenty of space between the rim of the plate and the inoculation from where you start. Okay, nowhere you should touch the rim of the plate. That is very important. At least 0.5 centimeter difference you always keep. Okay, so once you do, then with the loop you spread the sample like this, and then again you heat it, and again you spread like this. So at least 3 times if you can do, that will be well and good. The idea is at the end of the day you should able to get the isolated colonies of the organism. Okay, so if you are using the 2 samples in a plate, so like this it should be, they should not get contaminated or mixed together or anything of that sort. So when sample is plenty, so use full plate rather than a single plate. Okay, so this is how it looks like. So the inoculation should go first blood agar plate, then McConkey agar plate, then gram stain, AFP stain, or whatever is other stains that is required as per that. If it is a fluid, always centrifuge it, take the sediment. If it is sputum sample, try to get the sputum which the sample which is looks very good, not from the saliva, and you take. If required, you may have to homogenize it. So always ask the HOD when you are in confusion or anything. Okay, so these are all some other methods that is followed in other labs. So like this also you can do. Okay, this is also another, but at the end of the day what we want is an isolated colony of the organism. Okay, so this is how the 2 organisms have been put and the isolated colonies are got. Okay, in case of Kled agar plate, what you do, again you divide the Kled agar plate into 2 parts. Okay, so you put a straight line, you dip the inoculation loop into the urine properly, mix it well, and then with one single line like this you put. Okay, now do not heat the inoculation loop again. Instead you spread this sample itself. Okay, so like this you spread it. Okay, so if you are putting 2 or dividing the plate into 2, so then it should look like this. Okay, so whenever turbid urine is there, you can always use single plate. So don't try to use to divide the plate. If it is not looking very turbid, then you can divide the plate into 2. Okay, so at the end you should able to get this is also slightly heavy, but you should able to get a single colonies at the edges. Okay, so once that is done, make sure that in the respective microbiology book the entries are done, the lab number, the barcode, what is the sample, from where it is coming, and who is the doctor. So this many details should be entered. Okay, so along with this I will also make sure that you understand the how to handle blood culture positivity also. Whenever a blood culture comes positive, so from the blood culture always make a gram stain and check whenever you do a gram stain from the blood culture bottle, always mention that it is the gram stain is from the bottle, because otherwise which one is the sample and which one is the gram stain from the bottle, we may not be able to make it. So the word bottle should be present on the gram stain slide. Okay, so if it is a daytime, you wait for me to tell you whether it is a gram positive or a gram negative organism. If it is a gram positive organism, so you put a lawn culture on an MH plate for vancomycins and put the vancomycin, cefoxitin, clindamycin, erythromycin, amikacin, ciprofloxacin, amoxiclav antibiotic disc in it, and keep it in the incubator at 37 degrees centigrade. If it is a GNB antibiotics, again put one MH plate and put the following antibiotics: amikacin, ciprofloxacin, amoxiclav, polymyxin, meropenem, ceftazidime and ceftazidime clavulanic acid combination. If it happens in the night where we don't know which organism has grown and all, so during that time you put the combination like vancomycin, cefoxitin, then ciprofloxacin, amikacin, and ceftazidime, clavulanic acid, and amoxiclav. These many antibiotics you should able to put. Okay, hope the initial part you are able to understand. Thank you.